All right, so here is where we left off from the previous uh, video. So we left off going over the initiation of secondary growth in trees. So I did discuss how the vascular cambium is the layer that adds the additional secondary xylem and secondary phloem that gives the tree the width. So here in this younger part of the tree, you can see that vascular cambium, and you can see the xylem and the phloem. As the tree gets older, it's adding layers of secondary xylem and secondary phloem. So that's increasing the width. The older part of the secondary xylem accumulates protective compounds like resins and gums, and the dark colored inner xylem is called a heartwood, whereas the lighter outer part of the xylem is called the sapwood. Another thing you can see is that in areas with seasonality, the vascular cambium will stop growing during either dry or cold seasons. When it's dry because of the lack of water, when it's cold in areas where there's snow or ice, it's also because of lack of water because the ice is not, I mean, the water is not available. And so what this results in is the annual tree rings with alternating um, early and late wood. You can also see dormancy. So the vascular cambium will stop growing for a portion of each year. Okay. And this takes place during winter in cold climates or during the dry season in tropical habitats. And you can look at the patterns in tree growth rings as indicators of wet years. Because if you have wide rings, that indicates a wet years. If you have narrow rings, those indicate drought years. Okay. So when you're studying the growth rings in trees, and that's called dendrochronology. So dendro for tree, chronology for time, and sort of a sub branch of that is called dendroecology. That's when you're looking at not only the tree rings, but you're also looking at environmental factors that affect those growth rings. Okay, so here you can see the heartwood, the innermost part of the xylem in the darker color, and also the sapwood, outermost part of the xylem. And here at the bottom picture, you can see those tree rings. So when you have a cycle of wet years, you have thick growth rings, uh, and also and what you would see in dry years, it would be the thin tree rings. Okay. So it can be because of lack of water, or it can also just be other environmental factors. So in this case, they're showing the tree growth rings after the onset of acid rain and before the onset of acid rain. So before the acid rain, you have wide tree rings. After the acid rain, you have more narrow tree rings. And you would also see this in wet years and also dry years. Okay, so that's it for this PowerPoint. Now we're going to move on to the next one. So we are moving on to fungi. Let me just launch this thing. Okay, so fungi are eukaryotes that grow as either single cells or large branching networks of multicellular filaments. So some of the fungi that you might be familiar with are things like mushrooms, truffles, mold, yeast, okay, or mildews. And some of the important aspects of fungi is the way that they obtain energy, because there's fungi that are unlike plants, which make their own energy. Fungi obtain their energy from other organisms. So in that sense, they're more like animals than they are to plants. And so there are fungi that, um, well, they fungi can obtain their energy from both living and dead organisms. When they obtain their energy from dead organisms, they're playing a key role in environments because they are the only decomposers that can really efficiently decompose hard materials like uh, cellulose and lignin. So they play a key role in habitats. And then there's the fungi that get their energy from living organisms, like uh, by living in association with those organisms. Yeah. So those would be, you can see examples of mutualistic relationships, like the lichen, okay, like lichens, or when you see fungi living inside the guts of insects that and help them digest plant material, um, or like in the root system of many plants, where you have those mycorrhizal associations, where the fungi live within the roots. They take in, they obtain sugar from the plant, okay, because remember, sugar moves in both directions through the phloem of the plant. In exchange, the fungi provides the plant with the ability to obtain uh, water or nitrogen or phosphorus or other or any of those nutrients that the plant needs to grow. And so those would be examples of mutualistic relationships. And there's also parasitic relationships. So things like ringworm or athlete's foot, things like that, where the host uh, is just has its the fitness is being lowered on the host in exchange for a benefit to the parasite. Okay, so we're going to get into more details on all of those. So why do biologists study fungi? Well, they study it because fungi can cause a lot of debilitating diseases in humans and also in crops. Okay, so in crops, they can cause a lot of damage, which amounts to billions of dollars uh, in losses. And also because fungi are the ones that nourish plants that nourish us. So, like I mentioned, those symbiotic relationships with the mycorrhizal fungi. They also affect the climate change because they're critical to the carbon cycle on land. Oops. So there's about 300 species of fungi that cause human illness, and this is relatively low compared to other organisms. Fungi can also cause destructive impact on our food supply. So you'll get things like rust, smuts, mildews, wilts, and blights that cause billions of dollars in crop losses each year. You have the saprophytic fungi that are responsible for losses due to spoilage. So here's the parasitic fungi that infects corn and other crops. Although in some areas, like in parts of South America, they do eat these types of fungi as delicacies. But then there's also then you can see the ones that you've probably seen all the time. When you keep fruit too long in your refrigerator, usually this is what happens. You get fungi rot. So those were some of the negative impacts of fungi. Some of the positive impacts are that they are a source for many of the antibiotics, like penicillin. And also there's fungi that you eat, like mushrooms. Yeast is a fungi that's used to make bread, cheese, soy, beer, and other alcoholic drinks. Also, the chocolate, because fungi are the ones that ferment cacao seeds, and they make the chocolate edible. And then you have fungal enzymes that improve characteristics in foods, such as fruit juice, candy, and meat. The mycorrhizal fungi live in close associations with plant roots. These fungi along the roots are called mycorrhizae. So one question that you can ask is, would the plant growth suffer if mycorrhizal fungi were absent? And what they found is, yes, it does affect the plant growth. So here you can see uh, two examples. So you can see a plant that's grown with the mycorrhizal fungi and grown without it. So you can see there's a significant difference here. And here you can see what it would be, what you would see underground in the root system of the plant. So you would see the root of the plant, and then all of this lighter color would be the mycorrhizal network. So this is what allows the plant to obtain those nutrients that it needs, like nitrogen, phosphorus, and also it helps it absorb more water. Saprophytes are fungi that digest dead plant material. So these types of fungi help cycle carbons through the terrestrial systems. The carbon cycle on land has two basic components. There's the fixation of carbon by plants, and then there's the release of carbon dioxide by cellular respiration. And so for many of these carbon atoms, the saprophytic fungi connect the two components. So here on top, you can see the carbon cycle. So here's where the carbon atoms are being fixed during photosynthesis. Okay? And those atoms are stored in plant bodies, both living and dead. And then you have the fungi that digest tough materials like lignin and cellulose to obtain carbon containing nutrients. So they break the lignin and cellulose, and this allows other plants to use those nutrients that are held. So either plants or animals. So those carbon containing nutrients are returned to the carbon cycle, and then the carbon atoms are released again during cellular respiration by plants and other organisms like bacteria. So how do biologists study fungi? So there's about 110,000 species of fungi that have been described and named, but each year there's hundreds that are described each year. And as many as 6 million species of fungi may be found worldwide. For example, in one study, over 650 fungal species were found in the guts of 27 beetle species, and 200 of those species um, had not been described before. So now looking at some of the morphological traits of fungi, fungi have simple bodies. They have two growth forms that exist. There's the single-celled forms like yeast, and then there's the multicellular filamentous forms like mycelia or mycelium for singular. And you have some species that adopt both of these forms. Okay, so here you can see some single-celled fungi like yeast, And here's an example of the web-like bodies of mycelia. So all mycelia are dynamic. And so with mycelia, what they do is they constantly grow in the direction of food sources, and they will dial back in areas where the food is running out. The body shape of a fungus can change almost continuously throughout its life. And so we have hyphae, which are long, narrow filaments of mycelium. Okay, so when you have a lot of hyphae that forms a connective network, that's mycelium. Okay? So mycelium would be like if you have a, I guess you could say like a ball of yarn. If you pull a string out of that ball of yarn, that would be like an example of a hyphae. And hyphae are filaments that are divided into cells by septa. Septas are cross walls. And you have pores that allow materials to flow between those compartments. And some fungi are cenocytic, meaning that they lack the septa. And many nuclei are scattered throughout the mycelium. But the nutrients can move rapidly through the septal pores or through the cenocytic fungi from uptake to growth areas. Okay, so here you can see both the reproductive structure and mycelium are composed of hyphae. Okay, so hyphae, you can see hyphae in every part of the fungi. And here you can see those septa that are dividing the hyphae into compartments. And then where the cenocytic hyphae and the cenocytic hyphae, which lack the septal walls, you can see those multinucleate cells. Okay, so we'll pause here on this video and then we'll continue on the next one.